Next, we have a competitor returning to us from last year, Mathavarajah. He's a biochemistry and molecular biology student from Zosky College. This year, he's talking about uncovering the mysteries of Batten disease through an amoeba.
So we're all machines. We're composed of gears and tools that allow us to function the way we do. These gears and tools, biologically, we call them proteins. Proteins, they do a number of cool things in our body. They can act as scissors, they can act as glue, they can act as transporters, whatever it is, you name it, they can do it. When they don't work, we run into problems, right? And the disease I study is Batten disease. It affects children and adults, but nobody really knows how it happens, and there's no cure for the disease. So our lab focuses on trying to figure this out. And we focus on one of the proteins associated with the disease, CLN5. So far, up to date, nobody has had any luck figuring out what the function of CLN5 is. And this is mainly because isolating the protein has not been an easy job. Humans are complex, and so researchers, to kind of avoid that issue, they use model organisms. We know chimpanzees are related to us because they're similar and they behave similar to us, but we have distant relatives in many forms. And deep in the soil, we have Dictyostelium discoideum. I mean, it's not the cutest organism, but it's really cool. Dictyostelium discoideum has a protein which is similar to the human CLN5 that we have. And for, just to make it easier to say, Dictyostelium CLN5 is interesting to us, right? Because we can't isolate human CLN5, but maybe we can isolate Dictyostelium CLN5. That's the idea behind this, right? So if I can isolate Dictyostelium CLN5, maybe I can study the function of it and relate it back to human CLN5 and maybe figure out what's going on. So we did some cool experiments. I'm just going to show like the main one. And in this experiment, chemical X, I mean, it's intimidating if I give out the name. Just call it chemical X. And chemical X is bound to a sugar. Simple design. And the cool thing about chemical X is when the sugar is gone from chemical X, it begins to glow. So if it begins to glow when the sugar is gone, let's say CLN5 functions as a scissor. It comes in and cuts the sugar off. We should see it glow, right, in chemical X. That's what we see with Dictyostelium CLN5, which was really cool. And then we took it a step further and we isolated the human protein after some rigorous work, and then we saw the same thing. So through this analysis, we were able to determine that the protein functions like a scissor. It cuts these specific sugars off proteins. And so using this data, we were able to streamline exactly the function of the protein. And hopefully in the future, this helps us find therapeutics. Thank you for listening to my presentation.
So the runner-up prize goes to, I believe he was referred to as Sabi.